for this great introduction. Thanks for this gracious introduction, and it's definitely a pleasure to connect back with you guys. I spent like a number of years actually at ETH and UZH, so I was in the MD PhD program, which was UZH based, but then my lab was actually affiliated with ETH. So in the end, like throughout like my different courses, it was always switching back and forth between the two. And our PhD program, the international program in neuroscience, the ZNZ, is actually affiliated with both. So definitely great times. And I am very pleased that there's an initiative like the NEVEL community, which you guys have put together. There was nothing like that when I was a PhD student. And usually we used our hangouts and our hikes and our beer at the Heritable bar. Those were our outlets to talk about mental health, but I'm very glad that there is something a lot more organized and empowering right now. I will start with a very interesting literary anecdote here. Maybe some of you have read this book by Brian Weiss. It's called Many Lives, Many Masters. Now, it's a very interesting book. It's not like, I would say, purely scientific. There is, of course, like a lot of philosophy, a lot of speculation, may I dare say, even a little bit of pseudoscience, because it's about the story of like a young patient, a young lady who goes to see a psychiatrist, the psychiatrist being Brian Weiss back in the States. And then she's suffering from anxiety, which is inconsolable and not amenable to any kind of treatments. And then Brian Weiss, after like trying different modalities, he finally switches to hypnotherapy, and then starts traveling with his patient to her past lives. And it turns out that while she did not have any significant stressors or conflicts or traumas in her current life, in the past life, there were definitely a lot of experiences which have been, let's say, very impactful in a negative way on her life. And then from each life, it seemed like she acquired something, sometimes things which could give a strength to her personality, but also things which could add to the traumas. And then eventually Brian Weiss kept on resolving those traumas and like helping with shaping the perspectives. He actually learned a lot of perspectives from her past life stories as well. And eventually the patient gets cured with this past life hypnotherapy. And this is the account which is presented in this book called Many Lives, Many Masters. There's a very peculiar statement, phrase from the book, which I always refer to, which says that we are not sent from each plane with equal powers. Some of us possess powers greater than others because they have been accrued from other times. Now, I truly believe that whatever is written over here is true. However, perhaps it's not many lives which leave the impact, but it's whatever we acquire from our ancestors. And that is what epigenetics is all about, and what I'll talk over the course of the next 1 hour. So, when we talk about acquisition of behavior, the acquisition of traits from parents, we cannot forget that actually this was pointed out to us like centuries ago by Jean-Pierre Lamarck, who actually proposed that changes, adaptations, which we have to do to fit in with the environment, actually leave their mark on our code, what we call as genome now, and can be acquired by our descendants. And this is something which is present in many different species. However, the idea never flourished so much because there was the conflicting theory from Darwin. And at the same time, there was also this, I would say like a very strong hypothesis from another scientist named as Weismann, called the Weismann barrier, who actually like about 150 years ago, he proposed that, germ cells, which carry the code for the whole organism, they can transmit information to somatic cells, so the regular cells of the body, like the non-germ cells. And this is how the information transmission occurs. It's like one way from the germ cells to the somatic cells, but not the other way around. But because environments are affecting, our somatic cells first, right? That's how we perceive, we hear, or we listen, or we see, or we perceive. So they affect the somatic cells first, and there is no transmission which can take place from the somatic cells to the germ cells. So the combination of Darwinism and Weismann barrier effectively basically led to a demise of Lamarckism. And actually the reincarnation occurred decades later, more than a century later. When finally psychiatrists around the world, they started noticing this very strange phenomena that not only the Holocaust survivors themselves, it is intuitive to think because they went through a lot, were suffering from more neuropsychiatric illnesses than others, but even their children or even the grandchildren had more behavioral issues compared to, let's say, genetically similar controls who did not experience the Holocaust. Now, this, of course, became a topic of discussion because these studies were not mechanistic, they also met with a lot of speculation. And there were also a lot of articles which were published against this idea, questioning that can be really inherent trauma. Now, I will bring all of you to a very recent example. For many of you, I would say most of you actually, probably have seen this picture. So this is Sharbat Gul, known as the Afghan girl. She was photographed back in the 80s by, at that time, a relatively unknown photographer. And eventually the photo made to the cover of National Geographic. The agony on the face of this child over here then became a symbol of atrocities and traumas which children suffer around the globe during conflicts and wars. And again, a twist of fate that Sharbat Gul disappeared after that. And she was only discovered around about 25 years later. By that time, she had a child of her own. And also this photo of her with her child was very widely publicized. And a lot of people actually started saying that they can see the same kind of agony on the face of this little child over here, putting us into, this question that, is it actually that trauma can be inherited? Now, of course, this is just an anecdote, this is not a scientific study, but let's say if we start considering this idea that trauma of Sharbat Gul could be inherited by her child, then there are different possibilities of how this could happen, right? Number one, one could question that probably the mother and child, they were exposed to the same environment. And of course, in this case, Afghanistan has never recovered from the war which started 40 years ago. So perhaps the trauma just kept on happening. It was the same exposure in the mother and the child. Or it could be that there were other psychosocial factors which are responsible for the psychological effects on the child. It could be poverty, it could not only be trauma, but the factors which are associated with it, leading to it. It could be that just because of the war, as well as the atrocities and the social conditions, the mother was not nourished enough. And while she was lactating, she could not provide the right kind of nutrition to the child. And that is why the child now is suffering from any psychological illness. Again, there could be exposures during the pregnancy, which could be responsible for that. Or finally, there could be a germline dependent transmission. And that is what we call the epigenetic inheritance of trauma. There are different words, different terms used for it. You can also call it the intergenerational transmission of trauma. But that is the focus of my research. and perhaps a concept which has completely revolutionized the way, like, we think about biological heredity now. So this is like actually like a very simple graphic from a recent review article I wrote that got published in Trends in Genetics, where we looked at all the evidence. Like, it's a very comprehensive review, and we looked at hundreds of studies which could potentially be related to whether parental experiences or parental exposures could lead to specific effects, symptoms, or phenotypes in the children. And we found out that there is very strong evidence for transmission of psychological trauma and stress from parents to the offspring. And there is even evidence that there are effects at the germline level. So it means that trauma, which is perceived, initially by the brain, is leading to changes in the body which are captured by the germ cells and eventually lead to like phenotypes in the next generation. Similarly, there is very strong evidence about dietary exposures, high-fat diet or protein malnutrition, that these exposures in parents can also lead to metabolic perturbations in the children. But then there are also, I would say there's emerging evidence for also other factors such as old age or exposure to toxicants, and even sometimes positive exposures such as exercise. So there are studies where they have seen that, for example, there's a particular study where they looked at the fathers who were doing aerobic exercises before conception, and then they found out that there was like a lower risk of cardiovascular illnesses in the next generation. Now, these exposures, they lead to changes called epigenetic changes, which involves DNA methylation as well as RNA changes. I won't go too much into the scientific details of that, or wherever I have to, I will make sure that I simplify it for broader audience. So when we talk about the concept of epigenetic inheritance, what you have to consider is that the initial environmental insult, for example, trauma, it is affecting only the first generation, the founder, the parental generation. Now it leads to changes at the epigenetic level, so not the DNA sequence itself, but how it is expressed. And it produces a certain phenotype, so a certain kind of symptomatology, for example, anxiety, pro-depressive behaviors, or borderline personality disorder. So different kinds of behavioral features, they develop in the exposed generation. But here you would see that it is being passed on across generations, while the initial insult, the initial exposure, for example, trauma, is not there. So that is considered the hallmark of epigenetic inheritance. And I learned a lot about epigenetic inheritance during my PhD years and then postdoc years at ETH Zurich in the lab of Isabel Mansuy, where I worked on this model called MSUS. So it stands for unpredictable maternal stress and unpredictable maternal separation. This is a model of impaired maternal care. And it is, you would see that it is very recapitulative of a lot of different human conditions. So in this model, what we study is the effect of a dual hit, So one, these newly born pups, they are separated from their mothers in early postnatal life. And at the same time, the mother is exposed to this acute stress. So there is the combination of maternal separation and maternal stress, similar to what a lot of humans are experiencing around the globe. And when these mice, these little mice over here, when they grow to be adults, we see that they have a lot of different symptoms, ranging from neurobehavioral symptoms, but there are also changes at the metabolic level. There are changes in metabolites, and even we see that there are changes in the chemicals in the brain and in the circuitry in the brain. So it seems like this exposure is very prominent in terms of, let's say, impairing a lot of different mental and physical functions. But importantly, not only are these symptoms present in the exposed mice, but also in their offspring. Now, the key thing here is that the offspring is generated from one parent who's trauma exposed, but the other parent is naive, is wild type, is not exposed to any kind of trauma, but still the transmission is taking place. And going a little bit further into how this phenotype looks like, I could simply say that this is a very strong evidence that nurture can even affect the nature. So we all know that mice are nocturnal animals. They prefer staying in the dark. But what we actually see in these MSUS mice, as well as their offspring, is that they start spending a lot more time in the brightly lit compartment, which, if we try to interpret it in terms of mouse behaviorology, we would say that this is risk-taking behavior. So they are exposing themselves to an environment which is not in their nature. And again, what it shows is that this nurturing, this, I would say, negative nurturing through impairment of maternal care is really affecting the nature of an animal. What we also see is that they have problems in terms of we call it a pro-depressive behavior. So usually when you place like a mouse who's non-depressed in a glass of water, in this little beaker of water, you would see that they try to escape. They struggle. So they actually even manage to escape the platform. But what we see in these mice, as well as their offspring after trauma exposure, is that they keep on floating. So again, very similar to human conditions where in depression, people kind of give up sometimes, and this is exactly what we are seeing even at this animal level. Besides that, what we also see is that the social interactions in these mice, as well as their offspring, goes down. Their navigational skills are impaired, their spatial memory is impaired, as well as we see changes in their metabolism. So, when we talk about cholesterol, there are two different kinds of cholesterols. There is the low density lipoprotein, which is considered the bad cholesterol, and there's high density lipoprotein, HDL, which is considered the good cholesterol. So what we see is that in these mice, there is a reduction, a decrease in this good type of cholesterol. Now, importantly, we have to assess, or our aim is to find out what is exactly happening at the epigenetic level, which leads to this transmission to the offspring. Now, what I want to highlight is that there are changes in non-coding RNAs. So I will not go into, again, too much technical details, but non-coding RNAs are certain kind of molecules which are regulators of genomic activity. So they can go and control the expression of a particular gene. And by controlling the expression of a particular gene, they can alter the susceptibility to different diseases. So what we see in these trauma-exposed mice, as well as in the offspring, is that there are comparable changes in non-coding RNAs in the brain, blood, and sperm from the affected mice, as well as the brain of the offspring. So it seems also intuitive that trauma, which is initially perceived at the cognitive level in the brain, we see changes there, but then we also see changes in the blood, and we see changes in the germ cells. And then we see that through development of the offspring, they are being passed on. Now, what we did in our landmark study is that we took the RNA from the sperm of these trauma-exposed mice, and then we injected that RNA into our developing embryo. So the first one-cell embryo, which was considered from mating from unaffected mice, right? So we took the trauma-exposed material, introduced it into the unexposed material. And what we found out was that we still see changes at the behavioral level in the offspring, as well as the microRNA, the non-coding RNA changes in the brain. So it seems like the imprint, when it's present in the germline, if you take that imprint and if you introduce it into the unexposed developing offspring, you can introduce these changes. Now, we found out that there are changes at the germline level, at the germline RNA, but then it was important to see what is inducing those changes in the germline, right? And the connection between the brain and the germline is mainly the blood. So we had like another study, like, in which we took the serum, so a component of blood from these trauma exposed mice, and we introduced it, like, through repeatedly transfusions into naive mice, which did not have any exposure to trauma. And we again found out that this led to development of symptoms and phenotypes in the offspring of these mice, which had now been transfused with the MSUS serum. So again, what we now found out is that it is the blood-dependent factors which are leading to transmission of this trauma. Now, what I'm currently working on is a potential role for lipids, the fat molecules in this transmission. The very first reason I'm interested in that is because, maybe some of you would have heard of this, but both our brain as well as our germ cells, they are protected. They have these barriers. So there's a blood-brain barrier, as well as there is a blood germline barrier. And the only molecules which can freely go through these barriers are actually lipid molecules, which make them very likely mediators, of these effects of trauma. Now, another thing to consider is that, initially we talked about different exposures, positive exposure like exercise, but also negative exposures like high-fat diet, or trauma or stress or exposure to toxins, for which we now know that there could be intergenerational transmission. Importantly, all these different conditions, they actually affect the lipids in the body. So again, one more clue that lipids could be important. Now, another thing which is again hinting toward the involvement of lipids is that, we talked about HDL and LDL, right? The good and the bad cholesterol. And HDL, as we also showed, is decreased in this MSUS model. But besides its nutritional value, HDL is also a carrier of certain different non-coding RNAs. So these genomic regulators, which are present in different tissues. And what we have found out over the years is that the majority of these genomic regulators are actually present in the adipose tissue in the body, which means, again, that lipids seem to be the key. And not only are they present in the adipose tissue, they can be carried by HDL, and they can go from one tissue to another and alter the gene expression over there. So, maybe a few of you would have heard about this fatty liver disease, which usually in people who have like high cholesterol, they started having this sort of impairment of liver functions after many years. So it has been found out that actually in fatty liver disease, there are certain microRNAs, non-coding RNAs, which are released from this excessive fatty tissue, and they alter the expression of genes in the liver, and that is how the fatty liver disease develops. So again, there are clues from other medical conditions that lipids could be very important in this transmission. So this is what we are actually currently studying together with one of my graduate students. So we are focusing on lipids, their ability to carry these genomic regulators in the form of exosomes or HDLs or apoptotic bodies. Again, the details are not so important, but these are different packaging materials present in the circulation, and then their uptake through this HDL receptor SRB1. So what we are currently studying in our lab is not only early life trauma, but we are also looking at conditions which could either exacerbate the effects based on this lipid hypothesis or be preventive in terms of mitigating some of the intergenerational effects of early life trauma. So besides early life trauma, we also have models of high-fat diet as well as voluntary exercise. And we look at these effects at the epigenetic level. Now, because we talked about these lipids and their ability to transmit these genomic regulators, what we'll do is that after these exposures, we are going to sort of squeeze out these lipid-associated genomic regulators from one exposure and introduce them into mice without that exposure. So from MSUS, from early life trauma, we'll be able to show that lipid and their associated genomic regulators can transmit the signatures and affect in the same way the control mice. Or we can use high-fat diet and exacerbate the phenotype of MSUS at the intergenerational level, or we can use exercise and decrease the phenotypic transmission at the MSUS level. And finally, the advantage of mice is that you can genetically manipulate them and convincingly show which pathway is involved. That's the first step towards coming up with a therapy, which is then introduced in humans in the form of clinical trials. So for that, we are developing two different kind of mice. One mouse model is actually developed where we block the release of these genomic regulators in the adipose tissue, in the fatty tissue. So they have fatty tissue, but it will just not produce these genomic regulators, which could be like, transmitting the signals of traumatic conditions. And then we are developing these other mice in which, we are specifically targeting male mice in this because it's easy to access the germline. And I will explain to you in a minute, why it's relevant. And in these mice, in the reproductive tissue, the ability to uptake these lipid molecules, these HDL molecules, is decreased. So we either block the entry point or the release point. And through that, we'll be able to, these are two of my colleagues which are helping me out, we'll be able to convincingly show that lipids are important for this transmission, which then will bring us to potential therapies in humans. Now, when you want to translate this research into humans, it's very important to consider the difference between studying mice and studying humans, especially in a context of something as complicated as trauma. Now, number one, in mice, of course, we can look at the germ cells easily, but in humans, it's not easy to actually access the germline samples. Then there could be exposure differences because, of course, mice, we keep them in laboratory conditions, but humans, trauma never comes, I would say, in an isolated form. Usually, it's complex trauma, like people who have suffered, for example, paternal loss. A lot of times they are unfortunately also exposed to wars and genocides or abuse. And, so trauma can be quite complex. Then the question is how to have appropriate controls. There's also an importance of confounding influences because, again, trauma in humans can lead to certain risk-taking behaviors. For example, a lot of children who are traumatized, they eventually also start abusing drugs. So it's difficult to tease out which of the epigenetic changes are induced by drugs versus trauma. And of course, there is the element of genetic heterogeneity in humans. But we have actually done a couple of remarkable studies in which we had tried to navigate around some of these factors. So this is the consanguinity map of the world, So it shows how much cousin marriages are taking place in different countries around the world. And Pakistan actually happens to be the country with the highest rate of consanguinity. And there have been cousin marriages over the years. So if you would genotype, the population in Pakistan, they are more homogeneous. They are more closely related to each other compared to a Western population. Now, the only, let's say, advantage of that in terms of a scientific study is that we want to separate the effects of genetics versus epigenetics. So we want the genetics to be more or less similar so that we can isolate the effects which are happening at the epigenetic level. So that's why we performed the study in Pakistan, and we collaborated with the SOS Children's Village, a global organization who are hosting and nurturing children who unfortunately have lost their parents. And there we identified a cohort called paternal loss and maternal separation, PLMS. So this cohort is very similar to the MSUS model. Now, these are children who unfortunately lost their fathers, and the mothers, like, although they're alive, they're destitute, they're not financially viable enough to raise the children on their own. So they had to forcibly give the children for adoption to the SOS village. So there's the component of maternal separation as well as maternal stress, which is induced by the death of their spouses. So these children have this PLMS exposure, and we compared these children to their age, gender, and body mass index matched classmates. So again, we tried to adjust for as many of these confounding influences which we could. In fact, the study, this unique approach, it was actually featured in Science, one of the top scientific journals in the world. And again, like, they not only saw the scientific relevance of the study, but also the humanitarian aspect, which I want you guys to consider as well. So we have two different cohorts. We have PLMS children between the age of 7 to 12 years, both females and males. And then we also have an adult cohort, in which we only have males between the age of 18 to 25 years with previous exposure to PLMS during their childhood. What we found out was that the PLMS children, they are more likely to be depressed compared to controls, similar to the pro-depressive behavior which we see in the MSUS mice. We also see that these children, they unfortunately suffer from a lot of different anxiety conditions. And again, like, there is a bit of an overlap with the mice where we see social deficits as well as increased risk-taking behaviors. And also notably, even the metabolic phenotype seems to be conserved between mice and humans after childhood trauma, that we also see changes in the lipid levels of these PLMS children. Now, this was even a very surprising finding, because usually when you look at these children, they look malnourished. And you think that, okay, they are, they're more towards, let's say, the slim side. So we don't really intuitively think that they could have like cholesterol issues, but turns out that they do have cholesterol issues. And also then what we wanted to assess was whether there is a transmission of trauma, like, similar to the animals also at humans level. So which meant that we needed to have assessments at the germline level. So of course, like, in the PLMS children, we cannot do these assessments at the germline. So we collaborated with a diagnostic clinic, actually, also in Pakistan, where a lot of young men go for their reproductive health screening. So there we performed like a blinded analysis in which we took the semen samples, and then we also assessed these men for their childhood trauma using the childhood trauma questionnaire. And there we could then correlate the effects of childhood trauma to these epigenetic marks at the germline level. Here I would like to point out that because we were dealing with, like, previously only the PLMS exposure or the, let's say, parental loss or parental separation related exposure, we wanted to see if other kinds of traumas could also be leading to effects at the germline level. So we expanded, let's say, our repertoire of screening. And we found out that in this cohort, there were adults with different kinds of traumas, not only death of the parent or parental separation, but also traumatic sexual experience, physical victimization, severe illness or injury, or other severe traumatic experiences. Again, the cohort being from Pakistan, there were a lot of people who had been exposed to other kinds of victimizations or have been exposed to a terrorist attack, etc. So different kind of traumas were there. Then we also looked at the subjective severity. It turned out that traumatic sexual experiences have the most subjective severity, but all the other traumas more or less have the similar level of trauma severity. at the subjective level. So we divided our cohorts into 3 groups. So one without any significant trauma exposure before the age of 17, then CTQ1, which means that there was an isolated trauma, either of these categories, but just one event, or there was complex trauma, that there were at least 2 different events which these individuals unfortunately experienced before the age of 17. And then we performed sequencing for these genetic regulators. I will again not go too much into the technical jargon, but what I want to point out is together with one of my collaborators, we performed these sequencing for these genetic regulators. And you see a very remarkable effect of the trauma. So here we have, in this green line, these are the control children. And here we have the children with this paternal loss and maternal separation. And when we look at the expression of these genetic regulators through sequencing, the more red the dot is, the more, let's say, abnormally high regulation we see. Or the more blue the dot is, the more abnormally low expression we see. And here you see that there is a certain kind of clustering, that there are a lot of these different regulators which are overexpressed in the controls, or you could say underexpressed in the PLMS children. And then there are a lot of these regulators which are, you can see that there are a lot more pinkish and reddish in the PLMS group versus these children. So again, the effects are at a very massive level. Now, when we move to the sperm, here you see that there is not a clustering of these regulators, but more or less all these different kind of regulators, they are overexpressed in the group with complex trauma compared to the group with no trauma. I mean, there are some exceptions over here, but generally this part is a lot bluer than this part, which is a lot reddish or pinkish. Now that made us question that, okay, we see a pattern of all these different regulators either increased or decreased at the level of serum in the children, but at the level of sperm in these adults with childhood trauma, we see that more or less everything seems to be upregulated. So could it be that particular receptor, which is important for the upregulation, is altered? And that receptor could be the lipid receptor SRB1. And indeed, we found out that SRB1 was much more expressed in adults, in the sperm of adults with complex trauma exposures. So it seems that definitely lipids seem to be the key over here. And that actually has given us also a potential target to further validate in humans. So I would quickly mention a couple of more studies which we are doing at the lab in the laboratory right now, where we will use these different regulators that we found to be altered, alter them, at the germline level. And what we want to now assess is how long do they persist? So at which stage of embryonic development, let's say, could we intervene to prevent that the offspring does not have all these different pathological phenotypes? We also wanted to see if the effects which we see, could they be induced by any acute stressor? So for that, we took salivary, we checked for salivary cortisol levels in the PLMS children versus control children, and we found that there were no effects. So it seems like the epigenetic changes, they're induced by this strong traumatic exposure of PLMS, and not something, in which the child, let's say, was not feeling well, or there was some kind of a little stressful situation before, because the salivary cortisol was not altered between the two groups. What this very large analysis is also allowing us is to determine the regulators or the correlates of resilience versus susceptibility. Because what we have found out was that, when we look at children, so all the PLMS children were exposed to trauma, but then a number of children developed significant depression whereas some other children only showed very mild symptoms or no depression. So that allows us to also distinguish between the regulators, the epigenetic regulators of resilience versus susceptibility. And what we could do is that after fine-tuning this and after some further validations, we could introduce these resilience factors into the exposed group to enhance resilience. So this is also now allowing us to come to a particular therapy. One question which we have wondered and we have been asked about the role of lactation, that could it be not only germline dependent transmission, but could it be that women who were exposed to trauma during childhood or during pregnancy, for example, even, can they transmit any kind of genetic regulators which are then responsible for phenotype development of the offspring? So for that, we have another cohort. This is a cohort based in Poland, where we are doing milk assessments, as well as intergenerational neuropsychiatric assessments from families in which the mother was exposed to childhood trauma. And then we also have measures of whether they develop postpartum depression, etc. So that will also give us a lot more insight about this. And finally, I would like to mention the study. So a lot of you would have heard about the genocide or the massacre in Srebrenica back in the 90s. So we have also started a study at the familial level now in Bosnia, where we are comparing, familial transmission of trauma between the two cities, one which was massively affected, unfortunately, by the genocide, versus the city of Zenica, which was actually spared, where there was no genocide or no fatalities or casualties during the time of war. And there we again would be able to determine how much trauma transmission has taken place at the intergenerational level. And there we will also have paternal germline assessments, which we will then be able to correlate with the assessment of the children. So this is actually, I would say, something unprecedented and even sort of an upgrade on our previous study, where we looked at, where we assess children and then we assess the germline from adults who were exposed to trauma in childhood. But here we would actually have familial assessments, so we would be able to see the intergenerational transmission along with the germline analysis in the same family. So to put it all together, the topic I study is definitely, it's a challenging one, and it's also sometimes a very depressing one, because what we have found out is that trauma can alter lipid molecules as well as these genetic regulators. It can affect, the reproductive tissue and their ability to uptake lipid molecules and genetic regulators, leading to changes in germ cells, which affect children. And these are leading to pathologies in adulthood. And then certain dietary factors could even exacerbate that. But there are also windows of opportunities. And this is where I want us all to believe in science and believe in the propagation of knowledge, because by studying this, we are now also close to identifying potential therapies for this. For example, our current research may show that there could be a protective effect of exercise in this kind of transmission. By identifying the exact pathway of lipids and lipid receptors being affected, we now are getting close to a point where we could have targeted therapies, either through receptor blockade or employing the RNAs to then target these changes in the germline and prevent the transmission of this trauma. And finally, what we have also identified is that it's important at the clinical level, so for psychologists and psychiatrists, to not only take genetic family histories, which we already do. You go to a doctor and they ask you, whether your parents had diabetes or not. But I think what is also important is that we start taking epigenetic histories. We start taking histories of exposures in the parents, because that can also lead to susceptibilities. So there are, let's say, some positives like, which I want us all to focus on, because these windows of opportunities will prevent us from many of these scenarios to occur in future, where children have suffered and where potentially even generations of humans have suffered. So with that, I would like to wrap up. I'll happily take questions, but before that, I would just like to acknowledge my wonderful team who are working on it. My postdoc Isabella, my graduate students, Asha, Ismail, and Magdalena, as well as Patricia, our lab manager. We recently have had an expansion of the team as well. We have another graduate student joining us. And yeah, let's say this is our somber look, but sometimes we are also fun like that. So thank you, and I'll happily take any questions.
All right, thank you very much for this very interesting presentation, Adi. I guess somebody's clapping. I mean, I always find it very interesting to hear about your topic. It was like the second or third time, but I still found it fascinating, like what you're talking about. And I mean, I guess also, especially at the end, like when we're talking about potential therapies and stuff, it's definitely a topic where we can have a really huge impact. So it will be very fascinating to see what will come out of it. I guess before we start with the Q&A part, I just want to mention like 3 short aspects. So, I mean, I already talked about MeWell at the beginning, just in case any of you is interested in helping with organizing similar events, or also have like any other ideas, you're more than welcome to join. We're always looking for new members, and I guess that we can, you can either send us an email. I guess you can find the email address on our webpage, or you can also follow and contact us on our social media. Then second of all, it would be great if you could take the time to just fill out the feedback form for this event, just so that we can get get some kind of feedback and then see what was interesting, what we could improve on. So that's always important for us. And then lastly, I also want to mention the Speak Up ETH campaign, which is run by VSETH or VSETH, where students can anonymously send in testimonials about things that they have experienced in ETH environment, and therefore speak up about some kind of topic that is important to them. And they will then be supported by different contact points from ETH, and they will also receive a personal email response. So with that, I mean, you are more than welcome to ask any questions, and I will stop the recording then.